So I have a general question for you guys, and I really want to ask you this, and I expect you honestly to answer. Are you watching the recordings and going back to the lectures before you answering your assignment?
Of course.
Uh, so at least statistically, from Brightspace and YouTube, I don't get that number. So there is probably there are two categories of the students, one of them who really did and the other not. But statistically speaking, the number of hours of the YouTube and the Zoom and within the Brightspace doesn't reflect that there is 150 students who really logged in and checked the links. So that being said, again, I'm not in position to lecture you about what you have to do and when, but my role here is just to make sure that you're getting the right information and you have the access to all these information. You are good enough to know when you have to study and when you have to look for further references and all that. But I mean, midterm is closely approaching, almost like a week from now. So please, you also have to start looking into the material. And if you have issues, come back to me as quick as possible so that you don't end up at the end of the few days before the midterm where you have a problem. So first, let's go to the WooClap. Please go to your Brightspace. Let me make sure that the Brightspace is on. WooClap is on. Go to the WooClap, please, and then let's answer some questions. These questions would look like what you will get during the exam. So could you please, one of you, confirm that you can see the WooClap and that you can see the slide that shows the WooClap on your Zoom?
Yes, we can see it.
Okay, thank you. We will discuss on Thursday in details the questions of the midterm, and the assignment is due on Saturday midnight. 2 questions I got in there. Okay, so first question, which of the following statement about cell culture is true? Cell culture is only used for bacterial cells. Its primary cells are derived directly from tissues and typically have limited lifespan. Immortalized cells have a limited number of divisions and cannot proliferate indefinitely. Cells do not require sterile conditions as contaminations are rare. So a question like this, you need probably 30 seconds to get an answer. Twenty people answered. I need more to answer. You guys are around, how much we are on the Zoom today? We are around 66. 50% already answered. So, are you able to post all the work? Oh, Gary, I didn't understand your question. If you can't see all the question because it's long, just maximize the screen.
No, sorry, I meant like all the WooClap questions from previous lectures. Are you able to combine them in like a doc and then post them for review?
If you can see in each of the modules, there's one of those. I can definitely do that. It's easy, yeah. But there was a theme in each lecture. There was a question, so it will help you if you are reviewing that lecture. You will get questions similar to what you expect. Okay, so what is the correct answer? So 90% of you answered the question, so that's good. Primary cells, because we just take them out from the tissues, and typically they can't live for a long time. We need to immortalize them. So be careful, immortalized cells, they don't have a limited lifespan. Immortalized cells are like cancer, they can grow forever. Second question, what does a 260 to 280 ratio more than 1.8 indicate about a DNA sample? It could mean that the DNA is contaminated. It could mean the DNA is degraded and you can't use it. It could be the DNA sample contains significant RNA contamination. And it could mean that the DNA is highly pure. I can see that many of you are answering 3. I'm not sure is this the case for others, but I'm not able to see the work wrapping from the previous classes. Karen, I'm gonna look into that after this session today to make sure that all of them are visible for you. Okay. So as we discussed, when we have the 260 to 280, the 260 is the peak for the DNA and the 280 is the peak for the proteins. So when we have something at 1.8, it would look like that the sample is pure with minimal protein contamination. Now, some people answer 1, the DNA sample is highly contaminated with proteins. No, that's not true. It's slightly 1.8 and above. This means it's not contaminated with proteins. Now, the DNA is degraded. You can't know that because the degradation of the DNA is detected by the gel, not by the spectrophotometer. And number 4, the DNA sample contains significant RNA contamination. Also, this you cannot detect because when you read on the spectrophotometer, you just get a peak. You don't know if this is RNA or this is DNA. Okay. When we are doing primer design, which of the following is the most important? The primers should have a high content of GC, around 80%. The primers should have a melting temperature that is within 5 degrees. The primers should be at least 50 base pairs so that they assure the binding specificity. And the primers should contain long stretches of Gs or As to improve the binding. You have to be careful every time a multiple choice is being used. The multiple choices many times use some sort of your memory, like number 80, I'm sure you hear it's just like 80%. What does it mean, 40%? So there are some numbers related to the PCR that will come in those multiple choice, like 50 base pairs. 50 is like close to 55, which is the annealing temperature. And the right answer for this question is going to be number 2, because GC content should be 40 to 60%. 80%, this means it will have a very, very high temperature. We can't use it. The melting temperature between the 2 primers should be around 5 or within 5. More than that, the annealing temperature will not be favorable for this pair of primers. The primers should be at least 50 base pairs. That's not true. Primers are generally 20 to 25, and in certain cases, it could be more than that. And the more you add the Gs or As, you can probably make hairpins in the primer. So number 4 is not favorable too. The question about RFLP, what statement best describes the principle of restriction fragment length polymorphism, or RFLP? Is it to detect variations in DNA sequences using specific regions by PCR? Or is it to detect mutations directly by sequencing the genome? Or is it to detect the RNA expression in different tissues? And whether it is relying on the digestion of the DNA with restriction enzyme followed by fragment resolution by electrophoresis. I can't see the total number of people answering for some reason. So 4 people think that RFLP detect variations in DNA sequence by amplifying specific region using PCR. 1 person or 2% think it identifies mutations by directly sequencing the genome. So you have to be a bit careful when you are having a WOOC lab like this. Just for your knowledge, that are you answering because someone before you answered number 4, for example, correct? Or this is the choice that you would make in an exam. That's why we are having the WOOC lab now, to get flavor how much knowledge you still have or how much you remember. So it's important for you to think like on a multiple choice exam, how you eliminate possibilities that doesn't ring a bell. Like, for example, I'm very happy that nobody chose number 3 because it's RNA versus DNA. But RFLP identifies mutation by directly sequencing the genome. It doesn't sequence the genome, right? We don't have a sequencing step in RFLP. We didn't even discuss till today the sequencing of the genome. And first, RFLP detects variations in DNA sequence by amplifying specific regions using PCR. We don't have an amplification step before the RFLP, or at least we didn't discuss that modification of RLFP in our lecture. So the right answer should be 4, because you have to first take the DNA, digest it, run it on a gel, and compare the bands. The last question about the DNA fingerprinting. DNA fingerprinting uses the restriction enzymes to directly sequence the entire genome. DNA fingerprinting uses restriction enzymes to directly clone the entire genome, or it identifies unique pattern in individuals' DNA by analyzing short tandem repeats or variable number of tandem repeats. And finally, DNA fingerprinting is used to amplify specific DNA sequences through PCR to detect RNA expression. I don't see answering on this. Are you guys able to answer the question? Oh yeah, now it's 20, okay. 27. Again, these I many times repeat myself saying that those are anonymous. I don't have any grading on these. It's just for your own knowledge. I really encourage you to take the chance to answer, even if this was not 100% sure, because later on you will remember that you make a mistake here and you will choose the right answer. Okay, So just for the sake of the time, I'll give you like 10 more seconds. Okay. And the correct answer is DNA fingerprinting is the way we identify unique patterns in the DNA by using the STRs and the VNTRs. Now, DNA fingerprinting uses the restriction enzyme, but not to sequence the genome. We haven't gone to the sequencing. Be careful. You guys, you have to be careful for the wording. So DNA fingerprinting uses restriction enzymes, yes, but to directly sequence the entire genome, no, we didn't do sequencing the entire genome with the fingerprinting. And is it you using the restriction enzyme to clone the entire genome? No, because we are not generating a library here. And 4 is used to amplify specific DNA sequences by PCR to detect RNA expression. We haven't even discussed anything about RNA expression so far. And fingerprinting, we didn't really discuss the modifications that we have. Like there are too many details that I'm not really giving you, right? Because with this, I'm just giving you the basics. But if I dive more deep, I'm sure I will confuse you more. So I'm glad that the majority of you are answering the questions correct. For those who are not answering correct, please read the question carefully. And if you haven't gone through the material, please do. Okay. Now, one thing I would like to ask you, when you are reading the material, are you able to go to the YouTube links to see those techniques? Because I really think this is a very, very important component of the slides. Because if I describe for you a technique 100 times compared to the video that is showing you how it's happening, it will be much better, the YouTube, than what I'm saying. And I try to find, like, my role in this course is coordinate how I'm visiting those techniques back and forth. You can see that many times I'm going back and forth, so that I'm assuring that you guys understand what's happening. Now, one good thing I want to share with you, I wrote to the department, and I'm asking the department to provide a component of practical part for this course. And I'm in discussion with the dean to see how will this happen. I hope something shows up soon, and I will share with you the information. The idea is that some of these techniques, it would be much better for you to come for a visit somewhere in a lab or in some labs who agree to do that. And then you can see those techniques sometime, and you can ask a question about it, and it makes much more sense compared to all the theory that we are discussing here. Okay, so if we move on, are there any questions? Okay. So for today's lecture, we're gonna revisit a little bit what we have discussed before about vectors, plasmids, bacterial transformation, and how we can use those as a novel recombinant DNA technology in health biosciences. We will start now adding applications at the second part of each lecture. Because for now, I'm assuming that you have a good hold on how to culture cells, how to isolate DNA, how to isolate proteins. Later on, it's going to come an RNA. How you can get a DNA sequence, what's the use of restriction enzymes. So all the tools are now in hand. But now we'll start to put it in practice, little by little. So the first practice we will discuss today together is the DNA sequencing. And you will see that everything we described before about plasmids, bacteria, how to get a DNA sequence, how we can clone, how we can use a restriction enzyme, how we set up a PCR, how we make a primer. All those are component of DNA sequencing. And we add one more parameter on that, and we can do DNA sequencing with that. So let me first, okay. Computer froze. So let me refresh your memory again about the DNA application, because this is something that we learned a lot from it in order to be able to do a PCR. And it is also reflected on how we do the DNA sequencing. So if you remember, the DNA is being in a supercoiled condition in the nucleus. And when the replication has to start, we need a group of enzymes. And this group of enzymes will perform, each one of those will perform a certain function. For example, we first need to unwind the DNA. And in order to be able to do that, we have to ease the coiling of the DNA. So we need the topoisomerases to relax the supercoiling of the DNA strand. Otherwise, it will be like so difficult to reach into these interstrands. Then we will need the helicases in order to unwind the two strands. Only after those two steps, DNA polymerase come in and will be responsible for the synthesis of the DNA. Based on what? Based on the template. So each of these strands is a template. And therefore, once they are unwind, the polymerase is able to read that there's a T on this template strand, and its complementary part is going to be an A. So we have to have the nucleotides available, and we have to have the DNA polymerase and the template. And as I said, this is happening in our cells. So at 37 degrees, these enzymes will perfectly perform this function. Now, we use this concept in order to learn how to amplify DNA in vitro by PCR. And since we don't have those perfectly made enzymes to perform the steps, we will need to denature the DNA by heating it up to 95 degrees, and then bringing the temperature down to 30 to 50 degrees where the annealing of the primers take place. And then we will put up into 70 degrees where the 70 degrees where the polymerase works best, which is here at 72 degrees. And during this, at this temperature, the polymerase, if we have all the components, which are the optimized buffer that contains this, for example, MgCl2, and the polymerase itself, and the oligos or the primers where the polymerase need to look at the 3' end of the primer and start the synthesis, as well as the free DNTPs, and obviously the DNA template. So if we have all together, then the PCR reaction will work perfectly. So again, from DNA replication, we move one step to be able to amplify a small piece of DNA by PCR. Now, if we look a little bit further, we have to understand how the amplification step takes place. And for this process, where after the annealing of a primer on this side and on that side, well, definitely we have decided that between this sequence and that sequence, we are going to amplify a DNA. So you remember in assignment 2, I asked you, for example, amplify transcript 202. This means I already told you where is the start and where is the end of the transcript. It's already available there, so you know where are the primers. And your amplification is going to take place within these two primes. Okay. Now, for the amplification to take place, we have to remember some properties of the DNA compared to the RNA and the directionality of the DNA. So recall that the RNA has two OH group at the 2' and the 3' end, while the DNA has only one. That's why this is a ribose and this is a deoxyribose, right? Now, at the 5' end carbon, that's where normally we have the phosphates, the triple phosphates. And at 3' end, there's always this hydroxyl group on a DNA. Okay, so this is very important for us because whenever we are adding to a DNA additional bases, or in other words, polymerizing or synthesizing a DNA, we can only do that on the 3' part of the DNA. Because this, for example, this is a ribose, and the polymerization can take place somewhere here. This is not happening because that's more favorable anyway. But in the DNA, you don't even have that choice. So the positions 5' and 3' in the DNA dictate how the polymerization directionality take place. And during this step, a dehydration synthesis takes place where you get rid of this H group and then form the phosphodiester linkage. So bear in mind that always we have the synthesis from the 5' end toward the 3' end. Okay. Now, if we look carefully at the usage of a primer, and I'm 100% sure there will be a question on this slide in my exam. So DNA polymerization requires a primer. You cannot polymerize DNA without having a primer. Because as I said before, the polymerase needs to come and find 2 or 3 different aspects, at least 2 principal aspects. One of them, that the end of the primer has a free OH group that it can add this phosphate to the OH group. And it has to be able to read what is on the template strand and complement it with the right nitrogenous base. Okay? So if we don't have that OH group at the 3' end, the polymerase is not going to be able to perform the synthesis process. Okay? Now, when this happens, obviously we are losing these 2 phosphates, and we are adding a new nitrogenous base toward the 3' end, which it has, again, another free group. And the next one, if this was a C for the G, another one is going to be A complementary to the T, and the process is going to continue more and more. As much as the enzyme is active, the DNTPs are there, and the D buffer is suitable for the reaction to take place. Okay, so the directionality is also from this 5' towards 3' end. Now, taking all this information into consideration, the DNA replication, the PCR parameters, and the polymerization steps, then we can think about the sequencing as a very simple procedure where we could modify some of these nucleotides in a way that we don't get any recruitment of new base pairs anymore. And if we do that, then if we are synthesizing a strand on this side, and this C doesn't have an OH group, then the reaction is going to stop here. So that's the principle of this DNA sequencing. But how we do it and what are the parameters, we're going to discuss in the next slides. And the person who discovered this was Sanger, and he had 2 Nobel Prizes. One of them was for the ability to sequence the DNA. And his idea was exactly as I mentioned before: if you block the synthesis by using dideoxyribonucleosides, meaning there will be no more addition in the primer strand anymore. So he used those dideoxyribonucleotides, and obviously in the absence of the hydroxyl group at the 3' position of these, then there will be no more polymerization. And therefore, this will result in the synthesis termination. Now, how that can help us? So if you look up here, you have the DNTPs, the deoxy ATP, GTP, CTP, and TTP. You can see they have the OH group. And the ones that were used in the sequencing were dideoxyribonucleotides. So there's no OH group on those guys. Now, it's important to note that, and this is again, very, very important to remember that in the Sanger DNA sequencing, the nucleotides are mixed together. So you have the dideoxynucleotides plus the normal nucleotides. And in a reaction where you make the DNA sequencing, you want to have all the 4 DNTPs plus a lower quantity of that one dideoxynucleotide. And I'll describe this more and more for you a couple of times because probably you have never heard about this before. And again, we need the primer and the 3' end position so that the addition of these nucleotides will take place. So how does it look like? How does it look like? First, we have to find a way to be able to read the sequences. And by that time, in the 80s, they used to use the radioactive labeled nucleic acids. And for that, they used to use the phosphate P32 instead of the P33. And if you use a film, then the radioactivity can make you, allows you to detect the DNA. So this was the detection method. But if we look at a reaction, a normal reaction of sequencing, it would look like more or less a PCR. And if you look here, the sequencing reaction would contain a polymerase buffer, a DNA template, a primer, and DNTP. So these 5 components are in principle the same that you use in a PCR. Plus, on top of this, one radio-labeled dideoxynucleotide, only one. So if I have this piece of DNA that I want to sequence, then what I need to do is to get enough of that DNA that I can perform 4 different reactions. So I need to use, for example, 1 microgram of the DNA, let's say. So 1 microgram of the DNA template in each of those, plus all these 5 components go the same in all the tube, but the 6th component here, it's either ATP, dideoxy, ATP, dideoxy, TTP, dideoxy, CTP, or GTP. And it should be in a lower concentration compared to the total DNTPs. Why is that? Because you don't want to block all the reactions. You want the reaction to take place, and occasionally you want to stop it. So in other words, if we are reading here and all the ATP that I put is DATP, then every time I'm reading a sequence across this T, it will be a dideoxy ATP, and therefore the reaction will never continue. But if I have a mix of the dideoxy ATP and the regular deoxy ATP, then one time probably I get the deoxy ATP, and maybe the other time I will get this one there. So one time it will stop here, and one time it will stop there. So this is the basic concept how the DNA sequencing takes place. Okay, now, this is a very simple example for you. Imagine you want to sequence this 5' to 3' DNA. So this is the sequence to be sequenced. So the template of that, automatically it will be opposite, right? This is a 5' end, so this is the 3' complementary end, right? In blue, you have a primer. And I'm not going to discuss what primers we are using in the sequencing because that's more complicated for you. But at the end of that primer, then what do we have? We have the 3' end. Okay? And then whenever this is added, if this is an A, you have two possibilities. You have either the regular T or the dideoxy T. So if we imagine with the first polymerization, with the low concentration of ATP, the chance was the dideoxy is ATP was complementary to the A, we will end up sequencing only one nucleotide. And if we run that on a gel, it will fall all the way to the end as a T. Now, let's go to the second one. It's a G. So the complementary for that is a C, right? Then you have a possibility to have either the D-CTP or the dideoxy CTP, right? So if we end up having the dideoxy CTP in here, then the second is going to be a C. And now you know that the first sequence based on this band is a T, and then the second one is a C. Then go to the third one. It should be an A. Again, if it's dideoxy, it's going to stop there. And the sequence is going to be the third one as an A. So the idea is that we have 4 tubes. And sometimes for the A complementary, here it could be a T or dideoxy T. Here it could be a T or dideoxy T. And here again, T or dideoxy T. So and in this condition here, you will get 3 different bands for the T's. And the same for the others. Then we run each one of those on a gel and we'll make them run. And of course, the smallest is going to run the fastest and the farthest. And then we start reading from below. The first is a T, second is a C, third is a A, fourth is a G, and we start counting like this from bottom to top. And that's how we know the sequence. Now, there is a YouTube video for you to describe all the steps I said. Just let me see the time. So let's go watch this together. And oh wait, not this one. Probably we go to the second one. So in this situation here, we are using the radioactive material to be the method to label the DNA. But there's one issue is that we cannot mix those radioactive material together because then we will not be able to differentiate the bands coming from where. And also radioactive are dangerous. So scientists came back with new tags that are fluorescent. So instead of using the radioactive material, we now use a fluorescence. So we have the D, C, T, and A dideoxy that are fluorescent tagged. And we repeat the same thing we have described before, but now we can do it in the same tube because we can differentiate the different DNA molecules by their fluorescence. And the way we detect those is that we make those DNA fragments instead of going on the agarose gel to pass through a laser and then a detector. So the laser is going to hit those fluorescent tag, and then the detector is going to tell us this is a C, this is a T, this is a, this is a G, and it's going to give us the sequence based on their size. So again, starting from the smallest toward the biggest. And that's how we get what we call chromatogram that's generated, and it's going to tell us what is the DNA sequence. So let's stop here a second and quickly go and check the the video. My computer is acting very weird. Can't escape. Finally, I escaped. Okay. So let's go and watch this YouTube. Okay.
Order of the building blocks or base. DNA sequencing is the process of working out the order of the building blocks or bases in a strand of DNA. Before we can sequence the DNA, it has to be cut up into smaller pieces that are inserted into plasmid DNA and then put into bacterial cells. This makes it possible to produce lots and lots of copies of it as the bacterial cells multiply. The DNA is then isolated from the bacteria and sent for sequencing. The isolated DNA is transferred to a plate where the sequencing reaction will take place. A mixture of ingredients is added. These include free DNA bases A, C, G, T, DNA polymerase enzyme, and DNA primers. Modified DNA bases labeled with colored, fluorescent tags are also added. These are called terminator bases. To start the sequencing reaction, everything is heated to 96 degrees Celsius. This separates the DNA into two single strands. The temperature is then lowered to 50 degrees. This enables the DNA primers to bind to the plasmid DNA. The temperature is then increased to 60 degrees, and the enzyme DNA polymerase binds to the primer DNA. DNA polymerase starts making a new strand of DNA by adding unlabeled DNA bases to the target DNA. It continues to add DNA bases until a terminator base is added. These terminator bases have been chemically altered so that no more bases can be added to the new strand of DNA. Once a terminator base is added, the DNA polymerase enzyme stops making DNA and falls away from the strand. Everything is then heated to 96 degrees Celsius again to separate the new DNA strand from the original strand. This process of heating and cooling is repeated again and again to produce lots of fragments of DNA of different lengths. The length of each fragment depends on when a terminator base got added. To read the sequence of the DNA, the various fragments are separated by length using a process called electrophoresis. A capillary tube is lowered into each well of the plate and an electrical charge is applied. This causes the negatively charged DNA molecules to move through the capillary tube. Each capillary contains a porous gel. The shorter fragments of DNA move through the gel more easily than the longer DNA fragments. As a result, the fragments become arranged by size from the shortest to the longest. As the DNA fragments come to the end of the capillary, a laser makes the terminator bases light up. The color is detected by a camera and recorded. Each terminator base is labeled with a different color. A fluoresces green, C blue, G yellow, and T red. The shortest DNA fragments will be read first, and the longest read last. The sequencing machine records the color of the terminator bases as a series of colored blocks. Each colored block represents the labeled terminator base at the end of each fragment of DNA. By converting the colors into letters, we get the sequence of our piece of DNA.
Okay. So with the fluorescent
The human
tag,
body is made up of
Oops.
different tissues, each with a specialist function.
yeah, so nowadays we are not using anymore the classical sequencing. So this is the modern way to perform the sequencing using these fluorescent tags. And I hope this video showed you the steps are, and you can see that we moved one step ahead from a PCR to do a DNA sequencing. Similar concept, just one more parameter added. The other thing is that there is one modification that was mentioned in the YouTube about how the fragments are separated by electrophoresis with the application of the electric field. So this happens within the machine. You don't have to do it on a gel. And this reaction takes place within a couple of hours and you will get your sequencing back. And nowadays, we get all the sequencing done by the fluorescent tags. And these are some results that I have from some of the DNA I'm sequencing. It's important to keep in mind few parameters here. So if you look at the image here, that's how the machine is telling you that when that fragment of DNA had a terminator nucleotide with the red fluorescent, it was a T. So this is the peak that donates, it's a T. So it's very clear you can directly identify the sequence.
Professor,
Yes.
is it okay if you turn on your laser pointer?
Oh, yeah, sorry. Yeah, again. So I was saying that within the red sequence, you clearly see the peak denoting the T, the G, another T, another C. So there is a range where you can read very nicely the sequence. You have to be particularly careful in the beginning of the sequence, meaning the first 30 bases downstream the primer. Most of the time, the DNA polymerase, when it binds to those first 30 bases and start doing the polymerization, due to the annealing of the primer, due to the binding of the polymerase, most of the time we don't get a good reading. So that's how it looks like. And it's really specific. Like sometimes 29, 28, around 30, you start getting a very good reading. So if you need to sequence something, you have to keep in mind, you don't place your primer right before it. You have to give little space around 50 base pairs upstream of that so that you can avoid this noise here. And within the sequence, it's mostly good up to 1 kb, but above 1 kb, you start to get something like this. So you get, like, for example, there's 1 G peak, and there's a 3 that you don't know if they are really good peaks or not. 2 C peaks, 2 G peaks like this. And this is mostly happening more than 1 kb. So generally speaking, the primers can cover up to 1 kb maximum. So if you have a DNA sequence that is a 3 kb that you want to sequence, you might want to design 3 different primers. Okay, and the program to use for DNA sequencing is called ChromaSlide, and that's how it looks like. It's very easy and very friendly to use. So one thing that was mentioned in the beginning of the YouTube video about the DNA sequence, that it has been digested, cloned to a plasmid, and then this plasmid was transformed to bacteria, and then we amplify that, and then we isolate it, and then we do the sequencing. So would like to go now and describe for you the plasmids and the recombinant DNA technology, because that will give you the last piece of information we need in order to start using our DNA in experiments. So before, we discussed in module 4 how we make a genomic library, and we quickly use that as an example because we need to isolate DNA and we need to digest it with a restriction enzyme. And the other component in this was a plasmid or a vector that will also be digested with the same restriction enzyme. And then we will ligate those fragments into the plasmids and transform it to the bacteria. So today I will describe a little bit more about the vectors in general, about the process of ligation and formation of a transformant bacteria. Okay, so when we are generating the library, and probably this was mentioned before, I just like to add the text here because I looked at the slide and probably this text is important for you to know. If you take DNA and you digest it from a human cells, then there will be like a couple of million fragments. So that's a lot of fragments. And then if you clone those fragments into the plasmids, then you typically a clone, which is a one bacteria that has one piece of DNA in a plasmid in a bacteria, normally could have an overlap. So in other words, this fragment here could have a part from that fragment, depends on how restriction enzyme cut. And therefore, it's important later on to do any diagnostics for the plasmids that were generated and do the sequencing. And now we know how to do the sequencing. Now, just to go back one step into the previous slide, where it mentioned that you need to have a primer, right? Oops, where is it? You need to have a primer in order to start the synthesis of the DNA and the sequencing. And in this slide here, it's telling you that we are able now, because we know the plasmid we are using, so we are able to generate specific primers around the areas where, around the sites where these fragments are inserted, and therefore we can design a primer to go forward to read this way and another one to go the other way, reverse, so that we can cover the sequenced DNA very easily. Okay, now, generally speaking, we have more than one type of vectors. I only described for you the plasmid, and most probably I will describe only the plasmids for you. But it's important for you to know that if you hear in other courses or in other classes or somewhere else, plasmids are not the only vectors that we use, although there are many other naturally occurring molecules that we use. And the practicality of this vector is dictated by many things: how complex these vectors are, and how easy to manipulate them, and how easy to produce those or isolate them, and how much DNA sequence we can insert. So if you see plasmids are circular and can get up to 1, 5, 6, 7 kbs. Sometimes I did more than 7 kb in a plasmid and it still accommodate. But in case you're doing a genomic library and the sequences are really big, so probably you can't use the plasmids. You need something like BAC, which is a bacterial artificial chromosome or a yeast artificial chromosome. This slide is just to let you know that plasmids are not the only vectors we use. There are bacteriophages, for example, which are normally found in viruses. And these are genomes of viruses that infect a bacteria. And there are the cosmids, which are emerged between the plasmids and the phages. But for us, we are focusing on understanding the cloning and the recombinant DNA technology. If you apply it on plasmid, it's more or less applicable on others, but keep in mind that the size and the complexity can become different between these factors. Now, in a plasmid, we mentioned before that there are few parameters we have to take into consideration. In a plasmid, we need an origin of replication. And this is the element that allows the propagation or to copy the plasmid in a bacteria. Instead of having only one copy of this plasmid, you will have 5 or 10 or 20,000 copies. A plasmid contains an antibiotic-resistant gene. So this gene, when it's produced, the bacteria will become resistant to a certain antibiotic. And it's useful to have a unique restriction site, at least one, but many times with the engineered plasmids, we have multiple cloning sites, meaning more than one restriction enzyme site. And that's the restriction site here. And then we need to have a promoter. And as you know, promoters are used to induce the transcription initiation. And there are types of these promoters. Some of them are constitutive, so they are always on. And some of them are inducible, and these are more useful, in fact. If you want to induce the promoter, then you can have a regulatory effect. For example, you need a cytokine to turn on. You need a treatment with a drug to turn on the promoter. So whenever the drug is there, you can turn it on. And when the drug is not there, it will be off. This will come probably in the latest lectures when we discuss the protein expression. But it's important to know that there are more than one type of promoters that we can use. Now, we can modify these plasmids more and more. And just to go back one step, most of these parameters are naturally occurring in cells. Like bacteria had plasmids in order to get antibiotic resistance. So it's not us who devised this. The bacteria had found this way through evolution to become resistant to the antibiotic. And the same thing, the restriction enzyme was generated in bacteria because they used the restriction enzyme to digest the phage or the viral genomes. So this thing that I described for you here, the phage, it's the genome of a virus that is normally the restriction enzymes in bacteria would digest this in order to kill the virus so that the virus cannot overtake the bacteria. So again, this is a naturally occurring thing. So these plasmids nowadays, we played a lot with them. We played in the sequences. We generated a lot of variation of the sequences. So one of the modifications we added was to have more than one antibiotic. So in this plasmid, you have only one antibiotic resistance. In this plasmid here, you have ampicillin resistance and tetracycline resistance. And within ampicillin resistance, you have one restriction enzyme site, PSTL1. So this enzyme could cut through the ampicillin resistant gene. In other words, if I use this restriction enzyme to clone anything, if I cut through it, I will remove or terminate the effect of the ampicillin resistance, and the bacteria will have this plasmid, will not anymore have the ampicillin resistance. The same thing here, you have the BAMH1 within the tetracycline. So if I clone anything with BAMH1, I will interrupt the tetracycline resistance, and the bacteria doesn't have any more resistant to tetracycline. So we can use this in our DNA recombinant technology, and I will give you an example for that. So imagine you want to clone a specific gene, and you want to clone it to this vector, the PUC1890. This vector, as any other vector, has the replication origin, it has the resistant gene, and it has the LAGZ. Okay? But now we can add to the LAGZ. LAGZ stands for the beta-galactosidase gene. So it generates the enzyme beta-galactosidase. Okay? Now, if I want to identify whether a cloning happened in the multiple cloning site here or not, the first thing would happen if I would clone in the multiple cloning site and disrupt the LAGZ is I would stop generating the beta-galactosidase. And what is the function of the beta-galactosidase? How can I detect that? So if I would take beta-galactosidase enzyme and add the substrate which is called XGAL, then this will turn into a blue precipitate. The component that will be produced is 5-bromo-4-chloroindroxyl, and this has an insoluble blue color. Okay, so if the promoter is okay, LAGZ is being generated, therefore beta-galactosidase is being generated, and I provide on this plate here the component XGAL, which is the substrate that the beta-galactosidase use to generate this component that is blue insoluble, the bacteria will be blue. However, if I clone something and I disrupt LAGZ, what will happen? The beta-galactosidase is not going to be generated anymore. And therefore, even if I have the substrate, I will not have the blue color and the bacteria will be white. So it will be very easy later to detect where, in which of these bacteria I have a cloning that disrupted LAGZ and which one not. And then I can select only by the color of the colony. This is one of the fastest way you can select a colony that where you detect the cloning happened. Moreover, plasmids has been also generated in order to be propagated in bacteria to amplify them, to get a lot of these, as I said, because of the origin of replication. So you can see the origin of replication here. And the ampicillin resistance, so you can select which gene, which bacteria has that plasmid or not. On top of this, we could add promoters that work in mammalian cells. Some of these promoters, such as SV40, the cytomegalovirus, and here is the promoter of the simian virus as well. So these promoters allow the expression of certain genes in the mammalian cells. And in this particular example, the plasmid DNA3, pcDNA3, is one of the most famous plasmids. In any lab you go, it's found. We are trying to overexpress the GFP. So as you can see, the promoter is here, upstream of the gene. It turns on the GFP expression. It has a poly-A tail. Remember I mentioned about the poly-A tail for the messenger RNA stabilization. And therefore, whenever the promoter is on, it will transcribe this gene and the cells can turn green. On top of this, remember in the first class when we discussed about generation of cell lines. If we want to generate a cell line using such a plasmid, we can have the GFP positive cells because the GFP is being generated, green fluorescent protein being generated. And on top of that, we have a marker to select those genes. And this marker, for example, now is G418. So if we add this to the cells, only the cells that contain the plasmid with the resistance will stay alive and the others will die. And this is a way we can select quickly the cells that are transfected. And we can end up, if we are overexpressing MEK and RAS, those genes will transform the cell line into an immortalized cell line. And it will be a quick, easy, short way to generate a cell line. This particular part is coming in the next lecture again. So I will describe for you how we transfect mammalian cells and what are the types of the transfections. But I found it useful for you to know that this type of vectors is also used in mammalian cells. Now, how do we transform the bacteria? So this is the second component of the genomic library generation. It is a very easy and simple step where we simply incubate the bacteria with the DNA that we want, which is the plasmid in this case. And then there are two ways, either chemical transformation where we heat shock the bacteria. All the details are written for you here. And after that, we will put the bacteria in a selection media. For example, this bacteria doesn't have a plasmid that gives antibiotic toward ampicillin. This plasmid has the ampicillin resistant. So I will incubate together. I will do the heat shock, which is putting the bacteria from 37 degrees to 42 degrees for 30 seconds. And then some of the bacteria will get the plasmid. Those that got the plasmid, if I put them later on in a media that contain ampicillin, they will survive. All the ones that didn't get any plasmid, they will die. So that's the selection. The same thing can be done by electroporation, and it's by an electric shock. The only thing you have to be careful is that some bacteria are only competent for heat shock and chemical transformation. Others are suitable for electroporation. Normally, the highest competent cells are the ones that are used in electroporation. These are more expensive. And if you see here, I put you an example. The XL Gold strain is 27 times fold more more competent compared to the DH5 alpha. Okay. Now, in this slide, there is a video to show you the steps of the bacterial transformation, which I wrote most of them here. And please go and watch that. And there's another link to show you what are the growth media that we use for bacteria and how we can produce them. In case you want more information, go and check those. Okay. Again, my computer is freezing. Come on. I don't know what's going on today because my computer is like simply doesn't want to go for the next step. Oh, sorry. So the bacterial transformation steps are written extensively here, which is incubation of the bacteria with the solution of calcium chloride. Because calcium chloride is positively charged, it will get DNA closer to the bacteria and get the membrane stabilized. Then you heat shock. So this will allow the DNA to enter to the bacteria, and then you simply recover. So put to 37 degrees. And finally, you put the selection on the agar plate. Transformed cells are spread across agar plate containing the antibiotic. So only the ones that are transformed are in principally resistant. Okay. So with that, we kind of recover all the components of how we do a recombinant DNA, how we generate it from A to Z. Now we have it in a bacteria. But bear in mind that we are doing this in a lab, but nature has done this long time ago. So if you look at the corn that you're eating, it has nothing to do with the progenitors thousands of years ago. Similarly, with all the selections you see in dogs and in strains and in bacteria, up till the point where we started manipulating genes for introducing red fluorescent protein or green fluorescent protein in different animals and different models. So these steps we all learn from nature. And I just want to give you only one practical example of how we use the recombinant DNA technology. So the applications are really wild, could be industrial for generation of an enzyme or a hormone, and that's the example that I want to give you today. It could be clinical, and we can use all these to do sequencing, to identify disease-causing mutations, to identify SNPs, to check if there's an inheritable gene that causes a certain disease. All that under the clinical applications can be done, but the concepts are exactly the same. It's a DNA fragment, it's a sequencing, it's an alignment, and then checking all those back in addition to the basic research that we do. So the application of today is presented here for generation of insulin. So in principle, many people who have diabetes type 1 is mostly because of the loss of the insulin. And previously, people used to have the generation of the insulin in pigs. So you have to take the pig's blood, and most probably you are getting rid of big animals for that. Nowadays, what we can do is we can generate the insulin in a bacteria, in a bioreactor. The bioreactor, you can see it here, those big tanks, where they have a bacteria where we clone the insulin gene into a plasmid, and we have it selected by a antibiotic. And after the growth of that bacteria, then we isolate the insulin. I don't know if I have time to show you the steps of, and okay, let me bear in mind with me. So this is 2 minutes, please stay to watch. And in the next lecture, probably we can discuss more how insulin is being made.
Recombinant DNA technology can be used in the production of human insulin. Now, let's take a look at the production process. First, DNA encoding human insulin is obtained from a human pancreatic cell. Then, a restriction enzyme is used to cut the DNA. Meanwhile, a plasmid with antibiotic resistance genes is obtained from a bacterium. The plasmid is cut open using the same restriction enzyme. When the DNA fragments and the open plasmids are mixed together, some of the plasmids bind with the DNA fragments, and with the help of DNA ligase, they are joined together, forming recombinant plasmids. Note that some open plasmids do not pick up the DNA fragments and join by themselves. They are called non-recombinant plasmids. The plasmids are then mixed with the bacteria E. coli. This is to introduce the plasmids into the host cells. However, only some bacteria actually pick up the plasmids. These bacteria are said to be transformed, and only some of them have the recombinant plasmid. The remaining transformed bacteria have the non-recombinant plasmid. Meanwhile, some bacteria do not pick up any plasmids at all. In this example, the plasmid we use carry a resistance gene for the antibiotic ampicillin. So, to select the transformed bacteria, we first culture the bacteria on an agar plate containing ampicillin. the ampicillin resistance gene in the plasmid enables the transformed bacteria to survive. They then divide and form colonies. The bacteria that didn't pick up any plasmid cannot survive. After that, we transfer a portion of bacteria from each colony to a second agar plate containing another antibiotic tetracycline. As the transformed bacteria with a non-recombinant plasmid carry an intact tetracycline resistance gene, they survive and divide to form colonies. On the other hand, in the transformed bacteria with a recombinant plasmid, the DNA fragment inserted made the tetracycline resistance gene non-functional. Therefore, the bacteria cannot survive on the plate. From the results of these two agar plates, we can identify the colony formed by the transformed bacteria with a recombinant plasmid and select these bacteria for further processing. The transformed bacteria with a recombinant plasmid are then cultured in fermenters. In each bacterium, the recombinant plasmid replicates independently of the bacterial chromosome. When the bacterium divides, the plasmid is also copied to the daughter cells. As a result, many copies of the DNA encoding human insulin are produced. Finally, gene expression is induced in the bacteria to produce polypeptides. The polypeptides are extracted from the bacteria and processed into functional human insulin.